Okay, today we're going to start chapter 12, which is all about bacteria and viruses. And it's probably my favorite chapter in this in freshman biology. Bacteria and viruses are so cool, so fascinating and interesting, at the same time can be so devastating. so, um let's start with domain archaea. Real quick before we go into domain archaea, um, let's talk about this term right here, microbe. So we know that there is a whole world of things that we cannot see and that we were not able to see and we didn't even know was there until we invented the microscope. And remember, once they invented the microscope, everybody started playing with them, looking at anything and everything under the microscope, trying to figure out what is there that we cannot see. Now, anything that is considered microscopic, we call a microbe, and you do need to know that term and definition. And we know that what's crazy about these microbes is that even though we can't see them with our naked human eye, they have a huge impact on our day to day life. Look at what we're going through right now with the COVID-19 pandemic that's caused by a virus, this tiny little virus that we can't see with our human eye, is causing such disruption and chaos in our day-to-day life. So these microbes, even though we can't see them, they play a huge role in our lives. And so we're going to talk about bacteria and viruses in this chapter, but you need to know that term and definition. So we're going to start with talking about prokaryotes. And remember, a prokaryotic cell is one that there are no membranes around the organelles. And so both domain archaea and domain bacteria are considered prokaryotic. And that's where we're going to start. All right, with domain archaea, these guys are decomposers, but they are going to be found in extreme environments. This is your key term, red flag. You see this word extreme, you need to immediately start thinking archaea. So domain archaea are going to be found in extreme environments. They are prokaryotic organisms, although they do share some characteristics with eukaryotes. For instance, the way they replicate their RNA, the way they produce energy, how complex their polymerase molecules are. They have, some of these similar characteristics with eukaryotes, but they are considered prokaryotic because there are not membranes around their organelles, and you need to know that. So, domain archaea and domain bacteria used to be classified together. They all used to be one category called eubacteria, which is like all bacteria. But recently, scientists actually did divide them up because there were some big differences. So here are the differences between archaea and bacteria, and you need to know these. First of all, archaea are typically going to be found in extreme environments. Remember, this is that key word I told you to watch out for. Archaea are going to be found in extreme environments. And when we mean extreme, we're talking about places that you do not expect to find living things. The second thing is that the cell walls of archaea do not have peptidoglycan. So peptidoglycan is going to be found in the cell walls of bacteria, but not archaea. And peptidoglycan is what's going to cause a immune response in an organism. So when you are infected with bacteria, the peptidoglycan in the cell wall is what causes your immune system to go ahead and attack it. But that is not found in archaea. Instead, what we're going to see inside the cell walls of archaea is going to have isoprene. And this isoprene is this rubbery protein that's going to cause the archaea to become heat resistant. And so this is why you see archaea living in a lot of extreme environments. Places like volcanoes, deep sea hydrothermal vents, acidic springs, really super salty places where most of domain bacteria could not live. So in archaea, though this isoprene in the cell wall helps them to be able to live in those extreme environments. So those are two big differences. The third one is their transcription and translation processes are different. We're not going to go into that because I don't think any of y'all want to go back into protein synthesis. But just know that the way they go about making proteins are going to be different. So there's the three reasons why we split up domain archaea and domain bacteria, or kingdom archaea and kingdom bacteria. All right. Now let's talk more specifically about domain archaea. So we are talking archaea here. And there's four big types you need to know. And you need to know kind of where they live. First one would be thermophile. And when you see that word thermo and you think heat, you're right. Thermophiles are going to live where it is extremely hot, like a volcano or a deep sea hydrothermal vent, someplace where the temperature can go well above the boiling point of water. And in fact, these thermophiles need it to be hot. If the temperature goes too low, they will actually die. So thermophiles are going to be found where it is extremely hot and they need that temperature to stay up there. The second group would be the methanogens. One thing about this group, this is the largest group of archaea. So there's a lot of these guys. They're going to live in places where there's really none or not much oxygen. So we're going to find them in places like swamps. We're going to find them in the intestines of animals and humans. So yes, you can have archaea bacteria inside of your intestines. And in fact, what methanogens do is they produce a gas called methane. So within swamps, within the gut of animals and you have these little methanogens, these archaea, that are going to produce the gas called methane. The third group is called the halophile. These guys are going to be found where it is super salty, places that are saltier than the ocean. So Dead Sea, Great Lakes, salt mines, salt-cured ham, because that was using salt to preserve ham and other meats. You can find them there. So halophile is going to be found anywhere it is super, super salty. And then the last group would be the acidophiles. And of course, that's going to be found anywhere that's super acidic. So places like the acid springs of Yellowstone, things like that, where it is high acidity is where you'll find the acidophiles. Okay, and that's pretty much what you need to know about domain archaea. Extreme environments and really just knowing those four groups. Now, moving on to domain bacteria, we'll spend a little bit more time on domain bacteria because you're more likely to encounter somebody from this domain and this kingdom rather than archaea. In fact, you've got millions of bacteria all over your skin, inside your gut right now, and you need them. And remember, there's more good bacteria than bad bacteria. And so a few things to remember. We still are dealing with a prokaryotic organism, so there's no membrane around the organelles. These guys do act as decomposers. They'll break down dead materials and return it back to the environment. They will also help break down old cells, proteins, lipids on your skin and your intestines. So they do a good job of breaking stuff down. In fact, this is why you stink after you go to weight training or you go to sports practice. All that sweat on your skin is immediately being broken down by the bacteria on your skin, and it's that bacteria eating up the sweat that's causing you to stink. Okay. When it comes to bacteria, we typically recognize them and talk about them having one of three shapes. And you need to know these shapes. And in fact, these shapes oftentimes will be found in the name of a certain bacteria. So for those of you that might struggle with strep throat, like my son does every year I teach this, that's caused by the streptococcus bacteria. And so within the name streptococcus, it's telling you what the shape is. And so be aware that I could ask you a question I could give you a name of a bacteria, and based on which of these shapes is in the name, you would have to tell me what shape is that bacteria. And so here we go. The first one, the coccus shape, we say is an oval. And so again, those of you that struggle with strep throat and things like that, this is kind of what those bacteria would look like. It's more of an oval, kind of a circle shape. Spirillum is kind of what it sounds like. It is a spiral shape. And so you're going to see these guys kind of looking like this. So there's your spiral shapes. And the bacillus shape is what we call a rod shaped. To me, I think more like a pill, like, or a vitamin you take, but they say it's a rod shape. So it's kind of like that. So those are your three shapes. You do need to know those shapes. Now, it doesn't matter what the shape is. Bacteria are going to share similar structures. They have organelles, but remember, those organelles do not have a membrane around them because they're prokaryotic. Bacteria, domain bacteria, they do have peptidoglycan in their cell wall, and that is important. And in fact, what scientists like to do is they like to divide up bacteria into two groups based off of a technique called gram staining. Gram staining is all about finding out how much peptidoglycan is in the cell wall of that bacteria. And so based off of how much peptidoglycan is in the cell wall, they will classify a bacteria as either gram positive or gram negative. And I would write this down because this is important and you need to know the difference between the two. So what gram staining does is it takes the bacteria and it uses a violet colored dye, so like a purple colored dye, to stain them. And based off of how much peptidoglycan is in the cell wall, that bacteria is going to turn a certain color. So a gram positive bacteria is positive for peptidoglycan. It has lots and lots of the peptidoglycan. So because there is so much peptidoglycan inside this gram positive bacteria, that peptidoglycan is going to absorb the dye. And so if you are absorbing this violet color dye, a gram positive bacteria is going to turn purple. So those are good things to know. A gram-positive bacteria has a lot of peptidoglycan inside of the cell wall. Because of that, the peptidoglycan absorbs all of that violet dye during that process of gram staining, and gram-positive bacteria will turn purple. Now, gram-negative. You could say it's negative for peptidoglycan. It does not have much peptidoglycan. It has a lot less peptidoglycan than a gram positive. And so because this gram negative bacteria does not have much peptidoglycan in the cell wall, it does not really absorb the dye. And so in fact, these guys will kind of turn pink. So the gram negative, they only absorb a little bit of that dye because they don't have a lot of peptidoglycan in their cell wall. And so they actually turn a little bit of pink. Now, one reason why it's important to distinguish between gram positive and gram negative bacteria is because certain antibiotics do a better job of attacking and killing a bacteria. So knowing if you have a gram positive or gram negative bacteria is helpful so the doctor can know what antibiotic to help use to treat. And in fact, antibiotics, let's talk about that for a second, kind of get on a soapbox here. Antibiotics are going to be used for bacterial infections only. That is a question I could ask. Antibiotics do not treat viral infections. They're not going to attack or kill viruses. So if you end up with the flu or with COVID, they're not going to give you antibiotics to fight that. Now, there are times where you're fighting a virus that you end up getting a secondary bacterial infection, and you will need antibiotics for that. But antibiotics are not going to do anything for a viral infection. It is only for bacterial infections. And so the way an antibiotic works is it's either going to kill the bacteria outright, or it's going to do something to slow down the growth of that bacteria. Antibiotic could mess with the metabolism of that bacteria. It could not allow that bacteria to make the proteins or enzymes needed for that bacteria to function. Antibiotics could mess up the reproduction of the bacteria. Antibiotics could mess up the production of peptidoglycan in the cell wall. There's so many things, so many ways that an antibiotic can work against a bacteria. But the whole goal of an antibiotic, if it's not going to kill the bacteria outright, by slowing down the growth or messing up its function, is it's allowing your body, your own immune system, to have the upper hand. That way it can attack, and your own body will deal with and kill the bacteria itself. So antibiotics are there to help you fight these infections, and sometimes we need that little bit of a boost. Okay, let's see. Now, something else about bacteria, a lot of them do have capsules outside of their cell walls, and that's important. Notice it is outside of the cell wall, so this is like an extra layer of protection. And remember that a capsule is full of water, and so it keeps the little bacterium from drying out. It keeps it from dehydrating. But that capsule also makes the bacteria slippery. And so it makes it difficult for your immune cells to engulf it and capture and kill that bacteria. So that little slippery capsule helps the bacteria kind of squeeze and squirm its way away from your body's immune cells. So it's very helpful for the bacteria. Now, inside of your little bacteria, you do have DNA. We need that information to direct the activities of that cell. And you've seen the first one. You know the nucleoid. So here's your little bacteria with his little flagella. And we talked about like this kind of like mess of DNA right there. That is the nucleoid. And the nucleoid is made up of one circular chromosome. And actually what's interesting about both forms of DNA inside the bacteria is that they are both circles. They're both circular. And so the nucleoid, all of bacteria is going to have a nucleoid. They've got that, it looks like a mess of spaghetti in the middle there. And that's just the DNA. Now, some bacteria, not all, also, in addition to the nucleoid, are going to have something called a plasmid. And a plasmid is going to be another small circle. So it'd be kind of, like that, somewhere within the bacterium. And this kind of extra small circle, this extra piece of DNA is good for the bacteria, but bad for you. So what a plasmid typically does is plasmids are going to be passed between bacteria during reproduction. So it's information that they can pass along. But a lot of these plasmids have genes that help the bacteria during stressful situations. Well, what kind of stress could a bacteria be under? Well, you're taking antibiotics in order to defeat the bacterial infection inside of you. And so what these plasmids do, the genes on the plasmid can help tell that bacteria how to fight and win against the antibiotic. That way, you end up with what we have coined the term superbug. It's an antibiotic-resistant bacteria, and these bacteria are very, very difficult to deal with, and it's all because of these plasmids. Again, not all bacteria have it, but those that do, they're very resistant to antibiotics. They're very difficult to fight and get rid of. In fact, if you have ever heard of MRSA, it's a antibiotic-resistant staph infection, nasty skin infection. If you don't have a strong stomach, do not Google that. If you're curious and think you can handle it, then check it out later. But that's a nasty one that you see a lot of times in hospitals. But it's those plasmids with those genes telling the bacteria, hey, here's how you can get rid of, or here's how you can deal with the antibiotics that's trying to kill us right now. And then the last thing, a lot of bacteria are able to move using the flagellum. Remember, it's like this whip-like extension of the cytoskeleton that has a protein motor, and it's going to basically use like a propeller to help the bacteria move through its environment. Okay, so we're going to stop there. We'll come back tomorrow and talk about how bacteria can reproduce. There's a few ways and how they share information.